Okay students, we hereby start with the next part of the chapter, that is molecular basis of inheritance. Okay, in the previous part of the chapter we had discussed about how exactly the process of replication takes place. Okay, in the previous part we had discussed about what, how exactly the process of replication takes place. But in this particular video we are going to start with what, the experimental proof. Okay, experimental proof that the semi-conservative method of DNA replication takes place. Okay, experimental proof about what, the semi-conservative method of DNA replication. Okay, the DNA replication is taking place by means of what, a particular method called as semi-conservative method. Semi-conservative method. Okay, DNA replication is semi-conservative in nature, which was told by, or the DNA replication is semi-conservative, it was told by Watson and Crick. And how exactly the DNA replication takes place, about that we had discussed in our previous class or previous video, and we have ended up with what, we had ended up with the functions of DNA polymerase 1 and DNA polymerase 2 and DNA polymerase 3. Okay. Do remember that I repeat the same point by the beginning of this particular video. Okay. In the previous class we had discussed about the semi-conservative method of DNA replication, the process, and we at the last have said that DNA polymerase 1 and DNA polymerase 2, they actually do what, the process of proofreading and interchanging of the nucleotides or else nitrogenous base pairs. But actual process of polymerization is done by what, DNA polymerase 3. The addition of nucleotides will be done by what, DNA polymerase 3. DNA polymerase 1, what exactly it does? It will replace, okay, do remember, it will replace the RNA primers, okay, RNA primers with the help of what, DNA nitrogenous bases. Okay, RNA primers are replaced by DNA nitrogenous bases with the help of what, DNA polymerase 1. Okay, and DNA polymerase 2, this help for proofreading. And what about polymerase 3? It is the actual enzyme that helps for what, the purpose of, okay, it is the actual enzyme that helps for the purpose of polymerization, right. So, coming to the next part, that is Michelson and Stall's experiment. Okay, what is Michelson and Stall's experiment? It is the experiment that was given by the scientist Michelson and Stall, okay, in the year 1958. Okay, in the year 1958, Michelson and Stall have given this particular experiment, which proved that, okay, according to this experiment they have proved that DNA replication is, okay, semi-conservative. Okay, DNA replication is what, semi-conservative. Okay, it was just said that DNA replication is semi-conservative by whom? Okay, by the particular individual, that is Watson and Crick. But the experimental proof for semi-conservative method of DNA replication was given by, okay, Michelson and Stall. Okay. Okay, let us start with the Michelson and Stall's experiment. So, this is the experiment given by Michelson and Stall, okay, and it is the experimental proof for DNA replication is semi-conservative, which was given in the year 1958, okay, that we had discussed, right. And what they have done, how they have proved that DNA replication is semi-conservative, okay, very simple method they have used here, and that is they have grown okay, the bacterium, okay, they have grown the bacterium in the medium, okay, they have grown the bacterium cells or bacterial cells in a medium containing, okay, heavier isotope, okay, heavier isotope, okay, heavier isotope of what? Nitrogen, okay, they have grown the bacterium in what? Heavier isotope containing medium, okay, they have grown the bacterium in the medium containing heavier isotope of nitrogen, that is 15N, okay, 15N is heavier isotope and normal one is what? Okay, normal nitrogen will be having what? 14N, okay. First of all, what they have done? They have taken the heavier isotope of nitrogen in a medium and they have grown what? The bacterium in them. When the bacteria are developed in, okay, heavier isotope of nitrogen, okay, when the bacteria are developed in heavier isotope of nitrogen for several generations, they have developed their DNA, okay, with heavier nitrogen present in them, okay, they have developed their DNA with heavier nitrogen in them, okay. Then for several generations when they have grown the bacterium in the heavier isotope containing medium, both the strands of DNA of the upcoming bacteria were heavier, okay, because the nitrogenous base or nitrogen content whatever they have given is heavier for them. That is why all the DNA of those bacteria which were developed in heavier isotope containing medium, they have grown with what? The heavier type of what? Nitrogenous bases. That is why the DNA was having 15N in their nitrogenous base content, okay, 15N. Next what they have done? Very simple, they have transferred these bacteria, okay, they have transferred these bacterium in the medium containing normal, okay, in the medium containing normal nitrogen, that is 14N nitrogen, okay, 14N, okay. What they have done? First of all, they have developed the bacteria in heavier isotope containing medium, okay, heavier nitrogen medium they have grown it, okay. Later what they have grown? They have transferred those heavier nitrogen containing, okay, bacteria into the normal medium, that means the medium containing normal nitrogen. Then what will happen? As a result, okay, the DNA replication will be initiated here in the normal medium. Then both the strands will get separated, okay, the parental strands will get separated. On these parental strands what will happen? The formation of new strand will take place and the normal DNA which has got here, okay, the normal DNA which we do have here, okay, it will be having 14N and I will just use red color to indicate 14N. So new strands which are formed, they will be having 14N in them. Is that clear? They will have 14N. That is why these will be the daughter DNA molecules which are obtained, right. Later on what they have done? They have further grown it for the next generation, okay. Actually they have taken E. coli bacteria for their experiment, and when the E. coli bacteria were grown in the medium containing higher isotope of what? Heavier isotope of nitrogen, okay. All the E. coli bacteria have got heavier isotope in their DNA, so the DNA became heavier. Then what has happened? They have transferred that heavier DNA containing bacterial cells into a medium containing normal nitrogen, okay, into a medium containing normal nitrogen. At that time what will happen? Both the strands of the heavier DNA was separated and the normal DNA strands were formed on them. Next, in the third step, what have happened? They have allowed the same bacteria to grow in the same medium, that is regular nitrogen containing medium, for second generation. Then what will happen? As a result, the strands will get separated like this, okay. On these separated strands, see here, on these separated strands. Okay, these are the 4 separated strands obtained, and on these separated strands what will happen? The normal nitrogen containing DNA will be developed, right, like this. Okay. So, when it is developed, they have done centrifugation of the same. Okay. They have done the density gradient centrifugation. When they have done the density gradient centrifugation, then what has happened? In the first generation bacterium, okay, in the first generation bacterium, when they have done the centrifugation in the test tube, see here, if you consider this as the test tube, they have found the presence of DNA at the base of the test tube, okay, presence of DNA at the base of the test tube. But in the second, this is first generation, that means all the nitrogen were heavier, okay, all the nitrogen were what? Heavier in nature. In the second one, they have found that, okay, all of them were found to be in the intermediate region, okay. Why? Because in the second generation, whatever DNA are formed, here what has happened? One of the strand has got what? Okay, 14N, and one strand has got what? 15N. That is why intermediate region they have formed. Okay. In the third step, okay, that means in the second generation when they have obtained, they found that some of them were present at the surface, okay, and some of them were present in the middle, okay, and none of them were present in the bottom. When they have obtained 2 bands here, they concluded that some of them are present in the middle, means they are having 14N and 15N, okay, these 2, okay, they will form the band in the middle. And next one here, the lightest DNA will be formed, which will be made up of 14N and 14N, okay, these 2 are the molecules. That is why they have got 2 particular bands here, okay. Since they have got 2 particular bands in the third step, okay, it is very clear that, okay, it is very clear that DNA replication is semi-conservative, because only like this, if the strands are separated and new strands are formed on the parental DNA, then only we can get this type of what? Bandings, okay. Otherwise it is not possible. So here the major point you have to remember is, okay, see how exactly I will show you with the proper idea, proper clear idea, okay. See here, they have developed the DNA in a medium containing, okay, they have developed the DNA in a medium containing heavier isotope of nitrogen 15N. When they have developed the bacteria in the medium containing heavier isotope of nitrogen, then all of them were having heavier DNA molecule. They have developed a heavier DNA molecule like this, okay. Both of the DNA strands were having 15N and 15N in them, right. After several generations, when all the bacteria have got this particular heavier DNA, they have transferred these bacteria to a medium containing what? Okay, to a medium containing the lighter DNA, okay, to a medium containing lighter DNA. In this, okay, the medium was containing lighter DNA or else normal DNA, that is 14N, okay, it is 14N. When they have developed the same bacteria, or when they have transferred the bacteria containing heavier DNA to the medium having normal DNA or normal nitrogen, then what will happen? The both strands, okay, both strands will get separated first of all, okay, both strands of this heavier DNA will get separated like this, okay. Then after that what will happen? On them, okay, on these particular strands, the new strands will be formed, but new strand which is formed have got lighter DNA, okay. Why it has got lighter DNA? Because the nitrogenous base whatever present here is 14N. Okay, the nitrogen content is what? Having 14N in it. Okay, and the earlier one was having what? 15N. Okay, so here what has happened? These particular, okay, I'll write it here. These particular DNA which are formed in the new one, okay, they are what? Intermediate in their weight. See here, this is 15N and 15N, it is heavier in nature, okay, and here 15N and 14N, it is moderate in nature, and in the next generation if they allow the same thing, then what will happen? So the separation of these strands will take place, okay, and here also they will get separated, okay. I will show you how exactly they will be taking place. So separation of the strands, see, one strand here it is separated and other strand here it is separated, and this is also the strand which is separated, next here also another strand separated. So from this DNA 2 strands are separated and from this DNA 2 strands are separated. After that what will happen? The new strand will be formed on them, okay. New strand when it is formed, see here, then 2 types of DNA molecules will be produced, okay, 2 types of DNA molecules will be produced. So here see, it contains both 14N and 14N in it, okay. Here one 14N is there, another one is 15N, okay, and here also 14N and 14N, and here one 14N is there and one is 15N. So here what happens? This particular, okay, DNA molecules are moderate in their weight, but the first and third, these two are what? They are lighter in their weight. Yes or no? That is why in the first one when they do the centrifugation, okay, in the first one when they do the centrifugation, since it is heavier DNA, it will settle at the bottom. That is why it will be seen at the bottom. In the second one, if you observe properly, then what will happen? You can find the band in the middle. Why you can find the band in the middle like this? Because they are made up of what? Moderate amount of what? Or moderate heavy in nature. Okay, their weight is moderate. And in the third setup, you will get what? 2 particular bands, okay, 2 bands you will get, one is moderate and another one is light, because 2 different types of DNAs are produced here, one are moderate with 14N and 15N, okay, and others are 14N and 14N. That is why you will obtain 2 bands here. So this is of 14N and 14N, and it is of 14N and 15N, and here it is of 14N and 15N, and here what? The completely heavier DNA, that is 15N and 15N. So when they have obtained the result like this, they easily concluded that the DNA replication is semi-conservative, okay. DNA replication is what? Semi-conservative. Is that clear? So this is how they have proved that DNA replication is what? Semi-conservative. Clear? Coming to the next one, okay. Here is the diagrammatic representation, okay. Do remember, okay, do remember that in the first one, okay, the DNA was made completely heavy by means of developing the DNA in a medium containing heavier isotope of nitrogen, okay. When it is developed in the heavier isotope of nitrogen, all the DNA become heavier. Later on what has happened? It was transferred to the medium containing normal nitrogen or else lighter isotope or lighter nitrogen, okay, that is 14N. When it is transferred to 14N, then what will happen? The next bacterium will develop its own DNA. In that what will happen? Both of these strands will get separated. On the separated strands, new daughter strands will be produced, okay. And you know that here they have mentioned the minute, that is 20 minutes. The time they have mentioned is 20 minutes. Why? Because E. coli bacteria, okay, because E. coli bacteria will divide for every 20 minutes, okay. If it is going to divide for every 20 minutes, then what will happen? It is nothing but the DNA replication is completed in 20 minutes. Is that clear? So, next what they will do? They will transfer these particular bacteria after 40 minutes, okay, and they will check the content of the DNA, right. Then the result was obtained like this, and here first one was heavier DNA, second one was moderate or hybrid, and third containing what? Light DNA as well as hybrid DNA. So, by this process we can clearly tell that DNA replication is semi-conservative. Okay, by this method we can clearly tell that DNA replication is what? Semi-conservative. Clear? Okay. Coming to the next process or next part of the chapter, that is transcription. Okay, very important part of the chapter, okay, to remember, transcription. So, what exactly is meant by transcription? Okay, transcription is nothing but, okay, do remember, transcription is nothing but the process of, okay, I will write it here, the process of copying of genetic information, okay, genetic information, okay, the process of copying of genetic information from one strand, okay, from one strand of DNA into the mRNA, okay, the process of copying of genetic information from one strand of the DNA as a template or one strand of the DNA template into what? RNA. So, this particular process is called as transcription. Okay. And to remember, for the transcription, for the process of transcription, okay, the principle of complementarity, okay, the process of transcription, okay, will be working on the principle of complementarity. Is that clear? Or you can say that the process of transcription is regulated or governed by what? The principle of complementarity that occurs between what? Okay, the principle of complementarity that occurs between the nitrogenous bases. Okay, the complementary nature. What do you mean by complementary? So, A binds with T, okay, and G binds with C. At the same time, C can bind with G and T can bind with A. Okay, these are complementary base pairs. Okay, these are complementary base pairs. And here, in case of transcription, complementarity base pairing will be taking place. But the only difference is, since here the DNA, okay, information from one strand of the DNA is copied into RNA, what will happen? Instead of A binding to T, what will happen? A binds with U, okay, because in case of RNA, thymine will not be present, uracil will be present, so that in case the complementarity is working, okay, in the place of A, the next base pair which should come here? U should come here, because there is no particular thymine present in the what? RNA. Is that clear? In case of what? The particular RNA, the thymine is absent. Is that clear? And coming to this particular structure, okay, coming to the structure here. See, the structure which you can see in this particular slide, it is of what? The transcriptional unit, the structure of transcription unit. It looks very simple that only two, okay, lines are there and two boxes are there at the end, right? It looks very simple, but the thing is, it is very complicated, okay, because if only one point if you change, then the complete structure will be lost, okay. Always do remember, see here in the region they have mentioned 3 prime and 5 prime, and here it will be 3 prime and 5 prime, and this is the direction from left to right they have mentioned, and here they have written it as what? Promoter, is that clear? And here it is what? The terminator, okay, it is terminator. And next they have given structural genes and template strand, okay. Next the opposite one is called as coding strand, okay, coding strand, right. So if it looks very simple, okay, but it is most important to remember that, okay, if you are writing 3 prime end here, okay, and template strand here, okay, promoter region here, okay, if you write or if you write a particular point that is 3 prime and at your left side upper end, you have to take the promoter here only, and the direction should be from left to right itself, okay. Then on the opposite side you have to show what? The terminator, okay. If you change the polarity here, if you write 5 prime, then if you write 5 prime here, the promoter could not be this will become the terminator. That is why it is very important to note that if you write 3 prime and here, then next one should be what? Promoter region, and from left side to right side direction it will be taking place. That means the process of transcription will be taking place, okay. And the strand which is present here like this, it is called as a template strand. That means the coded information of this particular strand will be read. And what about other strand? It is called as coding strand, okay, whose information is not read, okay, the actual information read from the template strand, okay. Next, do remember at the end terminator will be present, okay, at the end terminator will be present. This is what? The transcription unit, right. So coming to the process of transcription, do remember that the transcription process take place in what? Okay, excuse me, the process of transcription takes place inside the nucleus of a cell. You have to remember, first of all, what is the transcription? The process of formation of mRNA from DNA, in simple words you can say. But if you want to clearly mention the definition of transcription, it is the process of copying of genetic information from one strand of the DNA template into RNA, okay. The process of copying of genetic information from one strand of the DNA template into RNA, okay. And where exactly the process of transcription takes place? The process of transcription takes place inside the nucleus of a cell, okay. The process of transcription takes place inside the nucleus of a cell, and this particular process of transcription, okay, takes place inside the nucleus with the help of what all? The enzymes, okay, the enzymes RNA polymerase, and you can call it as, okay, DNA dependent RNA polymerase, okay, you can call it as DNA dependent RNA polymerase. What exactly happens? How exactly the process will start? Okay, let us explain. The process of transcription takes place in the nucleus and it includes 3 steps. First one is initiation, second one is elongation, third one is what? Termination. The process of transcription takes place in the nucleus and the process of transcription includes 3 steps, okay, initiation, elongation and termination. What exactly happens here? The first step, initiation, it is the first step of transcription in which the process of formation of what? The mRNA begins, okay. What happens in this particular step of initiation? Okay, In the initiation step, okay, RNA polymerase, okay, the enzyme RNA polymerase along with the initiation factor called as sigma factor, okay. What happens in the initiation point or first point? RNA polymerase enzyme along with the initiation factor called as sigma factor, okay. Sigma factor is called as initiation, okay, initiation factor, right. RNA polymerase enzyme, okay, along with the sigma factor or initiation factor will come and bind to the promoter region of what? The DNA, okay. To the promoter region of the transcriptional unit or to the promoter region of DNA, what will happen? The RNA polymerase enzyme and sigma factor, along with the sigma factor, RNA polymerase enzyme will come and bind. So, this is called as initiation. I repeat the process once again. Initiation is the first step of what? The transcription, in which RNA polymerase enzyme along with the sigma factor, okay, or initiation factor will come and bind to the promoter region, okay, will come and bind to the promoter region, right. In the second step, elongation, okay, in the second step, elongation, what will happen? The particular polymerase and polymerase enzyme, the particular polymerase enzyme will move towards the right side direction, will move towards right side direction and ultimately start making what? The particular RNA, okay, starts making RNA based upon the complementarity basis, okay, based upon the complementarity nature, okay. The particular mRNA synthesis will be taking place here, see here, in the elongation step ಒಳಗೆ ಏನಾಗುತ್ತೆ, the polymerase enzyme, RNA polymerase enzyme go on moving towards its right side direction and slowly it start decoding, okay, decoding what? The particular codons or the particular nitrogenous bases that are present in the DNA, right. Ultimately what will happen? The mRNA will be produced by result, okay, mRNA will be produced, that itself is called as elongation. In the initiation what happens? Polymerase enzyme, okay, along with the sigma factor or initiation factor will come and bind to the promoter region, and in the elongation, okay, RNA will move towards RNA polymerase, okay, RNA polymerase enzyme will move towards the right side direction and slowly start producing the RNA molecule, which is called as mRNA, right. It slowly start producing mRNA molecule. And what happens in the third one? Okay, termination, okay. Third step is what? Termination. What is termination? End of the process here, okay, end of the process. Before going to the termination, I will just explain you how exactly elongation will take place. See, in this particular box I will explain you how exactly elongation will take place. So, if you consider this as the DNA, okay, if you consider this as the DNA, and here if the polymerase enzyme comes and attaches, this polymerase enzyme will open that particular region only, okay, it will open that particular region of the DNA and the nitrogenous bases will be made to undergo or made to what? Undergo unpairing. When the unpaired nitrogenous bases are formed here, slowly the polymerase enzyme which is attached to one particular strand, it will go on adding what? The nitrogenous bases, okay, or nucleotides according to the what? Complementary base pairing. According to the complementary base pairing, it will start adding or attaching the nitrogenous bases or nucleotides along with one another. So, here what will happen? A chain of mRNA will be produced, okay, chain of mRNA will be produced, and as the polymerase enzyme move towards its right side, what will happen? As the polymerase enzyme will move towards its right side, slowly the mRNA will get coming out of this one. See here in this particular diagram you can see the mRNA is coming out, and here the helix is open, the DNA helix is open only in the region where RNA polymerase is there, Okay, only in the region where RNA polymerase is there exactly what will be open? The DNA helix will be open for the decoding of this particular process. Is that clear? Ultimately, it reaches what? The termination. That is third step. In the termination, which is the third step, what exactly happens? Okay, do remember, termination is the last step of what? Transcription, in which the RNA polymerase enzyme will reach a particular region on the strand, on the template strand, where the rho factor is there or termination factor is there. When the polymerase enzyme approaches the rho factor or termination factor, okay, rho factor is also called as termination factor, okay, rho factor or termination factor. When it reaches the rho factor or termination factor, then what will happen? The RNA and polymerase enzyme and the DNA all together will get separated from each other. When all together are separated from each other, then it will lead to release of the RNA separately, clearly here like this, okay, release of the RNA, clear. Ultimately, what has happened? From one particular strand of the DNA, RNA molecule is being formed, and this process itself is called as what? The transcription. Okay, from one strand of the DNA, copying of genetic information, okay, has been taken place to produce RNA. This RNA is called as what? mRNA. Is that clear? This RNA which is produced is called as mRNA. And mRNA which is produced as a result of transcription, okay, will be also called as what? Okay, mRNA which is produced as a result of transcription will be in the form of hnRNA. Okay, will be in the form of hnRNA. So, what do you mean by hnRNA? Very importantly, in case of prokaryotes, okay, the RNA which is produced, it will be the final molecule, but in case of eukaryotes, hnRNA will be produced. What do you mean by hnRNA? Okay, it is the RNA produced by means of transcription in the eukaryotes. Okay, it is the RNA produced by means of transcription in the eukaryotes, and this particular RNA is called hnRNA because it will be containing, okay, it will be containing what? Two regions. Okay, it will be containing two regions. See here, it will be containing two regions, namely, okay, I am showing here, this is first region, right. It will be containing two regions like this. What these two regions are called as? Okay, you can easily distinguish between the colors I am using here, one is light and one is dark, right, like this. It will be containing two regions. So, these regions are called as what? These regions are called as, okay, these regions are called as introns and exons. Okay, this is intron again and this is exon again. Okay, introns and exons. So, what do you mean by introns and exons here? Very simple. Do remember, introns and exons are the different parts of what? mRNA. Since the different parts are there, the RNA which is produced is called as what? Heterogeneous RNA. Okay, HN is the short form of heterogeneous RNA. Is that clear? Heterogeneous. And what exactly is the use of these two? What is intron and what is exon? Very simple. Introns, do remember, are the non-coding parts of the RNA. Okay, introns are non-coding parts of the RNA, but what about exons? Okay, they are coding sequences of the RNA. Introns are the non-coding sequences, but exons are what? The coding sequences. Do remember that introns are non-coding, that is why introns should be removed. okay, because non-coding sequences are not used, okay, not useful, okay, it is like waste, okay, that is why introns should be removed. And what about exons? They are coding sequences, that is why they should be present alone. For that case, what happens in the eukaryotes, okay, for that sake, in case of eukaryotes, what happens is, okay, 3 steps will take place, okay. Whatever mRNA produced or whatever RNA produced, it is called as hnRNA because it will be containing what? Introns and exons. And these introns and exons, okay, that are present in the hnRNA, okay, here in these two, what will be there? The introns are non-coding, that is why introns should be removed, okay, introns should be what? Removed. So, first step, what will take place? The removal of, okay, removal of introns will take place. Removal of introns. What do you mean by removal of introns? Okay, the process of removal of introns, okay, is called as splicing. Okay, why the introns are removed by means of splicing? Because they are non-coding parts, okay, because they are non-coding. Since the introns are non-coding parts of the hnRNA, the introns are removed from what? The particular region, that is called as what? Splicing, okay, the removal of introns is called as splicing. And here in case of this particular diagram, you can see what has happened. Introns are there, okay, in the earlier one, but here the introns, see here, introns are removed by means of making a loop, okay. During this formation of loop, what exactly happens? I will show you, okay. See, during this loop formation, what happens? See, this is intron. Intron will be forming a structure like this. Loop like structure it will form, and by means of forming loop like structure, what happens? Okay, the exons will be attached end to end, okay. Here what will happen? Exons will get attached end to end, see here. Right, exons will get attached end to end. You can clearly see here, okay, clear ಆಗಿ ನಿಮಗೆ ಅರ್ಥ ಆಗ್ತದೆ ಏನಂತಂದ್ರೆ, here what has happened? Intron region is making what? A loop. When the intron region is making a loop like this, then at that time what will happen? The exon region, see here, exon regions are brought very near to each other and they are attached with one another. Is that clear? They are attached with one another. Like that, what will happen? Removal of the introns will be taking place, and this process of removal of introns is called as splicing, okay. The removal of introns is called as splicing, and this process of removal of introns will be, okay, taking place by means of forming the loop, and this particular loop formation will be taking place with the specialized proteins, okay. And when it forms the loop, the structure of this loop is called as what? Spliceosome. Okay, it is called as spliceosome. The proteins which bend and make the intron as a loop and ultimately removes it from the hnRNA. So this particular structure which is formed, it is called as spliceosome. By means of spliceosome formation, the removal of introns will take place, and the process is called splicing. Right. Next one, second step, okay. Whatever DNA, whatever mRNA which is obtained now by means of removal of the introns, it will be containing only the exons, that means only the coding sequence, right, which will be having 5' end and 3' end like this. But this particular mRNA which is formed, okay, by removal of what introns will have only the exons, but even though it is formed the stability of this particular molecule will be at stake. That means the stability will not be present. To give more stability to this mRNA or to this RNA, what we have to do? We have to carry out two particular processes, or else in the cell itself it will be making what? Or it will be undergoing two processes. Okay, the first one is capping. Okay, the first one is called as capping, and second one is called as tailing. Okay, second one is called as what? Tailing. Tailing and capping will be done. What do you mean by capping? Very simple. In case of capping, what? Methyl guanosine triphosphate. Okay, methyl guanosine triphosphate is added to 5' end of the mRNA. Okay, the process in which methyl guanosine triphosphate, if it is added to 5' end of the mRNA, it is called as capping. Okay, what exactly happens here? Methyl guanosine triphosphate. Do you remember methyl guanosine triphosphate? Okay, the process of addition of methyl guanosine triphosphate to the 5' end of the mRNA is called as what? Capping. The process of addition of methyl guanosine triphosphate to the 5' end of the what? mRNA is called as what? The capping. And second process is tailing. Okay, what exactly is tailing? I will write it here. See, tailing is nothing but addition of, okay, addition of, okay, several, okay, several, in general what? Around 200 to 300, okay, adenosine, okay, several adenosine residues, okay, several adenosine residues to what? 3' end of the mRNA. The process of addition of several adenosine residues, okay, the process of addition of what? Several adenosine residues. That means around 200 to 300 adenosine residues are added to the 3' end of the mRNA. So, that particular process of addition of what? Polyadenylate tail, or else the process of addition of the adenosine residues to the 3' end of the RNA. So, that will be called as what? The tailing. Okay. Ultimately, by means of formation of what, or by means of addition of methyl guanosine triphosphate and by means of addition of several adenosine residues, what will happen? Capping and tailing will be completed. After the process of capping and tailing, whatever mRNA is obtained, so that is called as mature mRNA. It is called as what? Mature mRNA. Okay. What is mature mRNA? It is the RNA which is obtained after what? The process of splicing, capping and tailing. After splicing, capping and tailing, whatever mRNA is obtained, it is called as mature mRNA, right. Okay students, let us continue with the next part of the chapter, that is genetic code. So, in the previous part we had discussed about what? The process of transcription. So, by means of transcription what we have obtained? We have obtained a particular mRNA, okay, which will be having the 5' and 3' end, a single stranded mRNA we have obtained, okay, by means of transcription. Then coming to the next part, that is genetic code. What exactly is meant by genetic code? Do remember that the mRNA which is formed by means of transcription, okay, this will be carrying the genetic information copied from the DNA, okay. The genetic information which is copied from the DNA will be present in the mRNA. and that genetic information which is copied from DNA, okay, and that is present in the mRNA, it will be in the form of what? Genetic code. That means the genetic information copied from the DNA, okay, will be in the form of coded part or coded sequence, right? That itself is called as genetic code. Okay, I repeat the point here. By means of transcription, what will be formed? Okay, the mRNA will be formed. Okay, that means of copying of what? Genetic information from DNA to RNA will take place by means of transcription. Okay, when the mRNA is formed, it will be having the coded information. Okay, the coded information from the DNA, and that coded information itself will be in the form of what? Genetic code, right? And this particular genetic code, okay, will be what? Triplet in nature. Genetic code will be triplet. That is the first point. Okay, the physicist by the name Gamow, okay, physicist by the name Gamow argued and suggested that combination of 3 bases specifies one amino acid on the, okay, particular mRNA. Okay, why the genetic code is triplet itself? Okay, if you ask this question, then the answer can be explained like this. So there are a total of 4 nitrogenous bases are there. Okay, they are adenosine, guanosine, cytosine and uracil. On the particular mRNA, 4 nitrogenous bases will be repeatedly occurring. That means out of these 4 nitrogenous bases only, how many amino acids should be produced? 20 amino acids. If one nitrogenous base is giving rise to one particular amino acid, then only what? 4 amino acids can be produced, but number of amino acid is what? 20, right? So genetic code cannot be singlet. Then combination of 2 if you take, okay, combination of 2, that is what? The combination of any of the 2 nitrogenous bases, that will be equal to what? 4 raised to 2, okay, because combination of 4, what combination of 2 you are taking, and totally how many number of nitrogenous bases are there? 4 nitrogenous bases are there. So if you take the combination of 2, it will be equal to 4 into 4, that will give rise to what? 16 nitrogenous 16 codons. But these 16 codons which are formed cannot code for all the 20 amino acids. Is that clear? Only 16 codons will be formed. So if you take the combination of 3, okay, that is 4 raised to 3, combination of 3, then what will happen? 4 cross 4 cross 4, it will be equal to 64. So in these 64 codons, okay, whatever we get 64 codons, in those 64 codons, casually or usually you can code for what? 20 amino acids. Is that clear? But more number of codons will be produced by means of combination of 3 here. But what will happen? Even though more number of codons are produced, okay, one particular codon may code for more of, or one particular amino acid, okay, sorry, one particular amino acid may be coded by more than one codon. So by means of that condition, we agree that genetic code is triplet. Okay, I will repeat the same point again. Do remember why genetic code is triplet? Because there are total of 4 nitrogenous bases present in the mRNA. They are adenosine, guanosine, cytosine and uracil. If 4 nitrogenous bases are repeatedly occurring on the mRNA, then what will happen? These 4 nitrogenous bases only will code for all the 20 amino acids which are present, right? And if all these nitrogenous bases, okay, in only one form or else singly they want to code for 20 amino acids, it is not possible, because if one is going to code for one amino acid, the 4 nitrogenous base can code maximum of what? 4 amino acids, but 20 amino acids should be coded now. For that, what will happen? We can guess that combination of 2 if it is possible or not. If you take the combination of 2, then total of how many nitrogenous bases are there? 4 nitrogenous bases. If you take the combination of 2, that will be equal to 4:2. That is 4 cross 4. That means 16 codons can be produced. In combination of 1, 4:1, only 4 codons can be produced. But in case of combination of 2, what will happen? 16 codons can be produced. But here again 4 amino acids cannot be coded, because out of 20, 16 can be coded, but another 4 will be left. So, that is why genetic code cannot become doublet also. If you take the combination of 3, that is 4:3, then it will be equal to 4 cross 4 cross 4. That will be equal to 64. So, in these 64 codons, if you obtain, you can code for all the 20 amino acids, yes or no? That is why it is confirmed that genetic code is a triplet. Okay, genetic code is what? A triplet. Then next point to be noted here is Hargobind Khorana, okay, Marshall Nirenberg, Ochow experimentally demonstrated the triplet nature of genetic code by using polyiodine nucleotides. Okay, polyiodine nucleotides. By using polyiodine nucleotides, okay, these particular scientists have experimentally demonstrated what? Genetic code is a triplet. And here in this particular box, okay, here in this particular box, you can see how exactly the combinations of, okay, triplets will be. Okay, how exactly the combination of triplets will be, and which all the triplet codons will code for which all the amino acids. Okay, see here, the first one, U and U here, the combination of 3 U, it will code for the amino acid phenylalanine. At the same time, UC will also code for what? Phenylalanine itself. And if you observe the next box, CUU, then CUC, CUA and CUG. Even though there are 4 different types of codons, what will happen? They will always code for only one type of amino acid. How it is possible and why it occurs? Even though they are different in nature, how can they code for the same amino acid? For this, another theory was explained, which is called as Wobble's hypothesis. Okay, that is called as Wobble's hypothesis. According to Wobble's hypothesis, okay, what happens is during the formation of a codon, combination of 3 nitrogenous bases will be there. But out of these 3 nitrogenous bases, say for example CUU and CUC. So, these two are what? A combination of 3 nitrogenous bases. But even though they are differing in the last base, they are coding for the same type of what? Amino acid, that is leucine. Okay, they are coding for what? The same amino acid, that is leucine, because according to Wobble's hypothesis, what happens? Only first and second nitrogenous bases which are present in the genetic code will determine which particular amino acid should be coded. Okay, and the last nitrogenous base which is present in the genetic code, it will not be affecting, or it will not be deciding the genetic, which it is not be deciding what? The amino acid that is to be coded. Okay. The deciding point will be present in the first and second nitrogenous base of the genetic code. That itself is called as what? Wobble's hypothesis. Wobbling of the codons will take place. Okay, wobbling will take place. That itself was described by whom? The Wobble's hypothesis. Clear? Okay. Next, coming to the next one, that is the salient features of genetic code. Okay. What are the salient features of genetic code that are present? Do remember salient features of genetic code. The first one is genetic code is a triplet. Okay, the codon is triplet. It means combination of 3. Codon is a triplet, and there are around, due to triplet nature of codons, there are totally of 64 codons are present, and out of these 64 codons, 61 codons are sense codons. That means they code for a particular amino acids. 61 codons are what? Sense codons, they code for particular amino acid, and other 3 codons are what? Stop codons. Out of 61 codon, out of 64 codons, 61 codons are sense codons. That means they will code for particular amino acid, but other 3 codons are what? Antisense codons or stop codons, which do not code for any amino acids. That do not code for any amino acid. And by the name, these 3 codons which are acting as stop codons are UAA, UAG, then UGA. These 3 are what? Stop codons. These 3 are stop codons. Next point. Some amino acids are coded by, some amino acids are coded by more than one codon. Hence, the code is said to be degenerate. That means a particular amino acid, say for example, leucine is coded by C, then CUU, CUU as well as the CUC, right. So, leucine here it is coded by 2 different codons, say for example, right. That means one particular amino acid is coded by more than one codon. So, that nature is called as degenerate nature of the genetic code. Next, at the same time, the third point, third salient feature, genetic code or codon is read in a contiguous fashion or continuous fashion. That means there will be no any punctuation mark like full stop, or there is no any punctuation mark like the comma. Right. Next one, the code is nearly universal. That means if AUG is coding for methionine in a particular individual, then say for example, in case of human beings, if AUG is coding for methionine, in all the organisms including bacterium, AUG will always code for methionine itself. That means if AUG is the codon, whether it may be present in humans or in the primitive organisms such as the bacteria, it will be coding for what? Methionine itself. Like that, it will be called as universal. That is, from bacteria to humans, what is there? UUU would code for phenylalanine as well as AUG will code for methionine, right. Next one, AUG has dual function. AUG has dual function. That means the codon AUG has got 2 functions. First one, it codes for the amino acid called as methionine, and it also acts as initiator codon. It acts as initiator codon. Do remember, it has got 2 functions. That is, the codon AUG has got 2 functions. That is, first one, it is going to act as initiator codon. First point. Second point, it is also responsible for coding what? The amino acid methionine. Clear? So, these are what the salient features of genetic code. Very important. Very important salient features of genetic code. And further we shall continue in the next point.